It's with my pleasure to introduce the last speaker of the morning, Dr. Erin Heinzen. I think it's fair to say that if it wasn't for the work of Erin and her colleagues at Duke, we wouldn't be here today talking about ATP1A3. Erin was involved intimately in the discovery of ATP1A3 as the genetic basis of AHC, and has continued to do really leading work in this area. Since leaving Duke last year, she's now at Columbia University in New York and is an assistant professor of the New Institute of Medicine. I hope I got all that right. But anyway, welcome to AHCF.
Thank you so much. Appreciate that. Can everybody hear me? Okay. All right. So yeah, thank you everybody for the invitation. This is really nice to be here. I was actually here back in 2011 when Kathy Swoboda and I were collecting patients and enrolling them into that. Ultimately led to the discovery of the gene. So it's really nice to be back and continue to share some of the work that we're doing at Columbia. So that's today to give just kind of an update on where we are with our research, and I'll talk specifically about one project and about the Epilepsy Genetics Initiative, and I'll explain what that is. But you know, I think there could be some value to letting you guys know about that work as well. I'm going to just give a very broad overview, so I'll be very broad, and then I'll try to get more specifics. So we are hoping that we'll sort of be the institute. So you know, we have a large focus on gene discovery. So we have a lot of human geneticists that are working to identify the genetic basis of disease. We have faculty that are focused on molecularly characterizing the mutations. And with the hope that that will ultimately lead to new therapies. So I think this is an approach that Dr. George and Dr. S also are working towards as well. And more detail on this. So you know, we are actively enrolling patients into precision medicine initiatives at Columbia. We've got focuses in epilepsy, neurodevelopmental disease, really any severe childhood diseases are being enrolled. We've also got kidney disease, liver disease, ALS, so lots of different diseases. We have a very large focus on neurodevelopmental diseases and epilepsy. So this is the workflow that we envision for epilepsy in general, but it's also very applicable to really any neurodevelopmental disease, particularly AHC, where there's also a lot of seizures and epilepsy. So we enroll these patients. They undergo sequencing. Very often we're able to identify the genetic basis of their disease. For a select number of patients, we'll then go into detailed molecular and functional characterization. And so a lot of the approaches that we're taking are very similar to Dr. George and Dr. S's approaches, but we essentially take our patient and we are working on developing a program for stem cells. Definitely not as developed as Dr. S's program, but this is ongoing work. We also have mouse geneticists who are taking the mutations and putting them into mice. So some people have brought up the CRISPR-Cas. So CRISPR-Cas has really made this pathway to developing mouse models much more efficient than it used to be. It used to take 2-3 years, and that's if things went well, you would ultimately have the mouse. You can now do it in, I believe, about 6 months. You know, if things go smoothly, you can get a mouse model of disease very quickly that you can then study. So we're specifically looking at brain pathology. So for our neurodevelopmental disorders, looking to see if the cells are in the right place, if the structure of the brain appears to be correct, those types of questions. Because we have a large focus in epilepsy, what we're doing detailed electrophysiological characterization. So this is the same as when a patient goes in, they get electrodes implanted and they monitor the seizures over time. We essentially are doing that in the mouse. These mice, it's not listed here, but behavioral analyses of just looking at how well the mice learn and attention. So what I'm going to focus on is actually this multi-electrode array, and I'll give you some more details. So we have a lot of work using this particular platform. It's I'll explain it in just a minute. But the hope is that using these models that we're able to then use it to screen compounds. And because we have these patient populations, many of them severely affected, it's possible that we can be able to initiate clinical trials if we were to find something in patient populations that we're collecting at Columbia. Okay, so the multi-electrode array, so just describing, the study, the electrophysiology of a single cell. So with the multi-electrode array, we can actually study the communication between cells. So the way this works is, so this is the system, but essentially each one of these wells is tiled with these electrodes. And what you do is you collect cells, you can plate them into these little wells. They then grow and develop on top of these electrodes, and then you can watch how cells fire. So we're not looking at the individual cell, like what's going on in an individual neuron. We're looking at how they talk to each other and how they interact with each other. So, yeah, as others have described, when one cell fires, it needs to propagate that to other cells in order to send its action potential. So that's we're trying to look at that communication aspect of the electrophysiology. So it's not going to give us as fine a detail as what Dr. George can elucidate from his single cells, but this communication piece I think is going to be really important. Let's see, so these are the cells. If you look really closely, you can see the electrodes with the cells layered on top. Okay, so this is in a single well. So each one of these is measuring what is picking up on cells firing. So each time you see one of these spikes, this is a cell firing. If you see these clusters, it's called a burst. So it's basically where you have one cell fires, and it fires kind of rapidly. And what you can see is that across all the electrodes, it seems to be picking up on this. So that means that the cells are, it's likely this electrical signal is being picked up across multiple cells. So this is telling us that they're kind of talking to each other. And we can use a whole bunch of different approaches that are being developed in the Institute to characterize this activity. So it's highly variable, very noisy. So we need to develop approaches to analyze these data to be able to pull out the real signal. Okay, so so this is not real data, just to let you know. So we'll try to figure that out. I'll explain in just a second. But essentially, these plots are taking all the data. So we've got mean firing rate. These are all the different parameters that we can pull from the data, and then we can plot. So if your red is a mutant neurons of some type, and your blue is what we call wild type or normal, you can look to see how they're different. So if we can understand how this communication is different, then we not only have a tool that we can use for drug screening. So can we give a drug that would bring this down to this point and essentially normalize that effect? But it also helps us understand sort of what's going wrong with the cells in terms of the communication aspect. So one of the sort of core projects that we have going on is to look at this D801 and mouse model. So this is the Mashalou, I think I'm saying that right, but this is the one that was developed by Mohammed Makati at Duke. So we have this mouse model, and we're using this approach to characterize the effect of the neurons with the intent of ultimately being able to screen compounds. We've also I think we have or are working on getting the D801Y so that we'd be able to compare directly. So now people have brought out that these actually seem to have a very similar mouse phenotype, but we believe that there's probably going to be differences because the majority of patients that have this mutation have a pretty distinctive phenotype compared to AHC patients. So we'd really like to see what that amino acid change difference translates to in the neurons. Okay, so this is the way we do this. So again, not real data. Don't try to interpret. So we collect immediately when the mouse is born. We extract the adult mice because we're developing neurons. So we take them right as they're born, what we call P0. So this is the day that they're born. We collect their brain tissue, dissect out what we call the progenitor cells. So the progenitor cells that ultimately develop into the brain and the neuron are located in sort of one region. We can collect those. make a little cell suspension. This then gets plated into these wells, and effectively the neurons develop in this plate. So almost like they would develop in the mouse to make the brain. They essentially just develop here in this plate, and we can study them on this multi-electrode array. This is just a snapshot of at first they're not very active. So these are just starting to develop, but you're starting to see a little bit of signal here. You can see the blue where it's starting to light up. This green could either be a real signal or it could just be sort of one of the electrodes kind of going crazy, because that's a lot of signal for that early on. And this is what it looks like over time. So on the first day, the neurons really aren't firing because they're still developing. So it takes a little bit of time, and you can start to see. This is just a sort of a generic plot, so it's not specifying exactly what we're measuring, but just to show you that over time this activity sort of kicks up, and then kind of it takes a little while to stabilize. Point, and this is where we want to study it. We don't want to study it really in this sort of dynamic period where we're not sure what's happening. We want to study it where it's nice and stable. And then, of course, doing the statistical test to analyze the signal. Okay, so this is just proof of principle. So this is, we want to make sure that this approach will work. So we have this. So Wayne Frankel has recently joined at Columbia. He's a mouse geneticist, and he has a cell4 mouse line. So this is this gene hasn't been implicated in human epilepsy yet, but we feel that it will likely over time become one, and we don't know that for sure. But there have been several examples where Wayne's been saying for years that this is an epilepsy gene, this is an epilepsy gene, but we never had any patient data to support that. And then over time, that's been brought to light. So we think that it's possible this could ultimately turn into an epilepsy gene. So this is the exact same, these mice. So the black is the normal, and the red is the mutant. As you can see, that even as early as day 4, day 5, we're starting to see that the mutant has a lot more signaling than the normal, and that's consistent with what you would expect to see in a mouse model of epilepsy. What's interesting is that there was data, kind of a long story, but there's some data from the mouse model that fluoxetine, a commonly used antidepressant, reduced. So just out of curiosity, we then treated with, so again, the black is the normal, red is the abnormal, and the green is the neurons that are treated with fluoxetine. And as you can see, it brings it back down to this baseline level. So that was an interesting validation, and shows us that if we can detect a signal from ATP1A3 mice, that we'll have a platform that we can study screen compounds, relatively high throughput, probably not as high throughput as some of the cell death assays or the other assay, which I can't remember, the flux assay that was talked about earlier. Okay, so now to get to some results, but don't overinterpret here. I'll explain. Okay, so this is the D801 mice. So this is relatively preliminary experiment where we have a relatively small set of mutant mice compared to the normal mice. And as you can see, we're not detecting much of an effect here. So these lines pretty much overlap. So these bars are what they call error bars. So it means like our certainty that the point is somewhere between this range. So as you can see, we're pretty much overlapping here. So there's basically three possible reasons why this could be occurring. So first is that there is no neuronal phenotype in terms of the communication. I think that's pretty unlikely given what we all know about ATP1A3 and neurons. But it's also possible that these cells are so high, even in the normal situation, they're sort of up at this upper level, and that if ATP1A3 mutants were even higher, you're essentially hitting the ceiling. You can't go any higher, so you're kind of stuck at the ceiling for both of these. That could be one reason. And it could also be that we have to challenge these neurons. So as many of the families and clinicians have taught me over the years, that very often patients require some, or they'll often have some sort of trigger. So it's possible that we need to stress these cells a little bit in order to see the effect. So that's sort of our next step, looking at that in more detail. This is where we are. Okay, so there's different approaches. I won't go into all the details, but there's different ways we can kind of stress the cells. So we can even just altering the ion composition inside and outside of the cell as a stress. So we can do that. We can also do it pharmacologically, just anything that will create stress. So we'll try several different approaches to sort of stress the cells a little bit and see if there's a difference. And there's also developing technology. So it's actually been around for a long time, but it's just now sort of becoming used readily in research. But it's optogenetic stimulation. So let's see if I can explain this correctly, but cells that exist in nature that are light sensitive. So you can put these into your cells, so you transfect them into your plated cells, and then you shine light on them, and that can, and there's two different forms. The channel rhodopsin, if you add the light, it will fire more, and if you do this other one, if you shine the light on it, it'll cause them to fire less. So essentially it gives you a tool where you can take the signal baseline. So we can either take it up or take it down. We're not sure which direction we need to go, but we can try both. And that allows us to try to dissect out the effects of the mutation. So if we are sort of at the ceiling, we can bring it down to where we can actually see the effect. Or if it's just that we need to activate it, to stress it in some way, then we can do that as well. So this is something that we're also working on in the lab. Okay, so I don't think I acknowledged her, but Melody, she's a graduate student in the IGM. David Goldstein is leading a lot of the work. He's also at the IGM. We're using these mouse models from Dr. McCarty and Karen Hartman as well. And we have to do this work. Okay, so that's sort of where we are with the functional. We'll talk a little bit about the Epilepsy Genetics Initiative. So this is a collaborative effort that's being led by the Cure Foundation, specifically Tracy, Dixon, Salazar. These are the scientific investigators that are involved. So these scientists have all been working together for years in the Epi4K consortium. So this is a large research effort to identify new genes in epilepsy. We have, for years, trying to find the genetic basis of the disease. But what we all sort of realize is that so many patients now clinically that what would really help is if we could bring all that data into a central database and actually be able to study. So the more samples we have, the better we can do in terms of identifying. So we're effectively going out to different physicians who routinely order a clinical exome effectively for an epilepsy patient, and it can be any epilepsy patient. The only criteria is that you have seizures. It can be older, younger. Anyone who's had clinical sequencing for epilepsy is eligible for the study. Data to be transferred in to this large repository that can be used for really two is direct benefit to the patient. So very often a patient will be sequenced clinically, but the diagnostic company that will give them a report that will say you have X, Y, and Z variants. We think we found it. And very often patients will receive either a negative or a non-specific, where it's what they call a variant of unknown significance, where they don't know what the mutation does. But of course, epilepsy genes are being discovered, multiple genes a year, because of the new technologies that we have. So it's really key that the exomes are analyzed and reanalyzed over time. And so that's one service that we'll do. So for all these patients, we'll take the current literature evidence, reanalyze, and then if we find something, we then let the doctor know that they need to get this site re-clinically confirmed so that the patient can be told. So it's sort of a much more dynamic process. So a lot of the companies will allow for a single reanalysis, and that's sort of included in the cost. But this is ongoing. So every 6 months, the patient's exomes are reanalyzed in the context of what's currently known in the literature. We also, of course, use to assemble this large cohort so that we can better identify genes. So we're also continuing our searches for new genes in this cohort, and we're also making the data available to any researcher who requests the data. So we've got about 120 patients in the database as of yet. We have lots of instances where we have confirmed what the clinical sequencing has found. So this is a patient who had a GRIN2B mutation, and it was identified on clinical sequencing. We also found it, so it's nice to know that we can find it as well. This is just showing the timing of when the publication was, or when the gene was discovered versus when the patient was sequenced. In this slide, it's actually kind of a little bit more of an interesting case. So this case really exemplifies why the importance of this sort of dynamic screening. So in this case, the patient was sequenced at the end of 2005. In early 2005, the publication came out reporting this gene. So if this patient had been sequenced in early 2015, they would have, it was only because actually 4-month difference. So we expect that that there will be a lot of instances where new diagnoses will come out because the patient was just sequenced too early. They just didn't have the evidence yet for that particular gene. We do have one case where we provided a new diagnosis. So in this case, the patient was sequenced, and typically the company sequence sort of the core exon, so for a gene, but sometimes there's novel exons that we don't know as much about. But the companies have to keep things to screen these sort of core exons. They reported that the patient didn't have any variants that were likely pathogenic. On our analysis, a mutation that was in a novel exon 5. We discussed with a lot of people who have done a lot of research on SCN and 8A and believe that this is truly a pathogenic mutation. So this is now in the process of being reported back to the patient through the physician. So the system guard. We haven't found any new genes yet, but we're hopeful with these now that we're really starting to get a lot of patients into this, that we'll identify new genes through this process. So right now it's limited to, well, let me just first say, so we're in the process of expanding that because we want more numbers. These are 8 sites, so these are essentially just sort of the rural sites, I guess, that were slated because they had a lot of patients coming in, getting exome sequencing, and the physicians were willing to sort of do a little bit extra work at the outset to get these patients enrolled in the study. But we are expanding, and it's also possible, so if an individual patient is interested, they can contact our counselor and get enrolled that way and put their data in for continual reanalysis. So our hope is that as we can sort of show that this works and is successful, that we'll expand, open this up so to diseases and include all AHC patients as well as many other neurodevelopmental diseases. Yep. So I think that's all I have. I had one quick thing that I didn't have slides on, but some of you have been asking about all the different mutations that have been found in patients. So that's something that we've noticed that we really need to compile. So Hendrik Rosewich and I are working on compiling those and making sure they get entered. So there actually is a database that's NIH funded. So it's the ClinVar database that essentially lists all. So researchers have to submit their mutations, but they give a little bit of phenotypic information. They often have contact information of who to write. If you want to get in touch with either the researchers, usually it's not directly to the families, but you can contact the researchers who can sometimes put you in contact with the families. But we're working on, so for all the different researchers who may have unpublished mutations, to try to gather them and submit them to that database, so at least it'll be up to date for everybody developing research models or families wanting to know if another child has that mutation, those types of things. That's it.
Hi. I have a quick question for you. Thank you for coming and explaining this. And I was just curious, when you're talking about the communication, that much variation between the mutated and the normal or wild type, if we assume that there's some sort of dysfunction in the cell, would it be communicating the problem, like, so the communication is fine, and but it's correctly communicating the problem versus a problem in the communication.
Right. Yep. So you're exactly right. So I mean, I think that's probably the case, and so that's why it's really nice that that Dr. Jordan will sell what can we read from that, and then what can we tell from the communication side. But I think very likely there's going to be like you said. I mean, the communication may be fine, normal signal, which might be better found in single cell model. But yeah, I think you're exactly right.
Okay, thank you.
If you're not part of the study of the epilepsy one where you're getting your genome sequencing, what should we be asking to have done? Like, should we, because it's moving at such a rapid pace, and 4 years ago and got a confirmation, how often should we be requesting that we get it resequenced?
In these cases, we're actually not resequencing, we're just reanalyzing the data that exists. But so I mean, if you received a negative report, I mean, I would contact the company who did ask them what their policy is on reanalysis. So sometimes they'll charge you, sometimes they'll do one free. It really depends on the company. So I mean, I would say that if you have a negative result, I would say maybe, just because it'll be a little bit of a tedious process and it'll probably take a while. I mean, 6 months would be ideal, but I'd say at least every year I would be trying to see if you can get that custody, given the most recent literature.
Okay, thank you.
Given that I think probably a lot of the people in this room, genetic data is with Dr. Swoboda. For us, what's the best way to approach being sure that we're in the initiative and that our data is ongoing and in the right place?
Yep. So I think that's something that Dr. Swoboda and I should work together on and see if it's possible. So we do require a special consent because this is a little bit broader than just sort of looking at just data into a big epilepsy database. So we often have to reconsent. But if that data is available and you want it in the database, then I think it's certainly an option. So that's something that she and I can discuss at some point. If there's a way to just, because so much of the data is in her database. The other alternative is that it's possible we could get her access to the EGI database, but I think there is value in sort of keeping it everything together in one central place, so that way, and it goes through the reanalysis pipeline. So most AHC patients have ATP1A3 mutation, but there are some who don't, and making sure that those patients have the best chance of getting a genetic diagnosis.
Great, thank you.
When you're doing epilepsy right now, but you got the AHC data.
Right.
The kids with AHC and epilepsy, do they have two mutations in, do you expect, or I mean, you're only analyzed for AHC.
Yeah, right.
Duke, right?
Right. So, well, I mean, we look at the entire exome. So, I mean, in theory, we should be able to detect both. But with epilepsy, it's a little bit harder of a question. So, because it's much more what they call genetically heterogeneous. So, lots of genes cause epilepsy, whereas one, possibly two or three, might cause AHC. So, it's much easier to find that. So, we don't know. It could be that they have a second mutation. It could be, a direct effect of the existing mutation. I think that's going to have to sort of unfold over time. And as we get as we get further along with the functional workup, or we get further along with gene discovery in epilepsy, because it's so heterogeneous and we need these massive sample sizes. If we can find new genes, then we can go into the AHC patients and say, do they have mutations in those genes that appear to be pathogenic? And that might, over time, help us sort of tease that out. But right now, we don't know. We're not sure. I mean, my gut instinct is that it's probably a direct effect of the ATP1A3 mutation manifestation of that, but we don't know that for sure.
Is there planning on taking that data from the previous studies on AHC alone and revisiting it for an epilepsy?
So that's kind of been off and on discussed in various forums. So I think for sure, especially now that most patients are sequenced, I think that's going to be a really critical next step. So I think we all the researchers who have genetic data do need to get together to do that, because we should be looking. Okay, all right, thank you.